Hello everyone. This video is about how to enumerate the aerial microflora with the help of settle plate technique. So first of all, I'll sanitize my hand with alcohol so that there is no contamination. So as you can see over here, we have already prepared the media plates and we have allowed it to solidify. So this is a plate wherein the media has solidified, and this is the plate that we will be using. It is a nutrient agar plate. So nutrient agar are basal media, that is a common media that is helpful for any kind of microorganism to grow. So we will be using this nutrient agar plate, and along with that we have one PDA. So we will be enumerating both bacteria and fungi from the air. So this one PDA, it is for fungal isolation and this is for bacterial isolation. So what is settle plate technique? It is also called as a gravity settle plate technique. So the aerial microflora or microorganisms that are present in air will be trapped onto this media. Let me open it slightly near the flame so that it does not get contaminated. So if you can see, this is solidified media. So we will be taking out these plates and we will be placing these plates at different locations. I am only giving you two examples, but we have multiple sets of such plates, and we place them at different locations wherever we want to enumerate the aerial microorganisms. Say for example, one can be in the lab, one can be in the corridor, one can be in the bathroom, washroom, or one can be in the kitchen, or one can be in the outside environment. So wherever we want to enumerate the presence of microbial load in air, or what are different types of microorganisms or bacteria or fungi present in air, we can use this settle plate technique for that process. So what happens in this process is, we place the plate and we remove the lid of it, and likewise we leave it for about half an hour. So due to their molecular weight, the microorganisms that are present in air will settle down and will get impregnated onto these plates, and after half an hour we cover the lids of these plates and we incubate them. The bacteria goes inside the incubator, that is incubated at 37 degrees centigrade, whereas the fungi is incubated outside the incubator in room temperature, and fungal cultures usually take more time. In bacterial plates we get growth after 24 hours, whereas in case of fungal culture we may need 72 hours to do even more for complete growth of fungal cells. So let us now see where we can place this. So this is the laboratory rack and this is nearby the incubator. We want to see if there is any microbial load, higher microbial load in the air. So for saying that, we will place it here and we will leave the lid open, and it will be left here like this for half an hour, and after half an hour we will close the lid and then we will incubate it. So this can be actually practiced after fumigation of laboratories and hospital rooms. Once we have fumigated, and even after that, if we have any microorganism left, that can be checked with the help of settle plate technique. And similarly, we have another plate that is of the fungi. So we will place the PDA, potato dextrose agar, or you can have SDA as well, that will also be placed over here, and we will leave it for half an hour and we will allow the microorganisms to settle, and after half an hour we will be putting it for incubation at room temperature. So it has already been half an hour now. We will cover the lid of both the plates. The fungal plate will be kept at room temperature, so we will leave it here. We will seal it and we will leave it here for incubation, and then this bacterial plate will go inside the incubator for incubation. Place it here for incubation, and tomorrow after 24 hours at 37 degrees centigrade incubation, we will see bacterial growth in here, and from there we can identify what are different bacterial cells or bacterial culture that are present in the air inside the lab.